This demonstration aims to image fluorescently tagged proteins in live cells in a 3D matrix. First, generate stable cell lines expressing fluorescently tagged proteins. Seed these cells into a 3D collagen matrix. Then acquire time-lapse images of migrating cells using a confocal microscope. Ultimately, results illuminate protein localization and dynamics within migrating cells through time-lapse imaging and FRAP analysis.
Observing protein dynamics and localization in 3D and in real time offers one avenue for addressing fundamental questions in cell migration. For example, how are adhesive contacts regulated by cytoplasmic proteins? And also, how are these contacts perturbed by specific inhibitors?
In a P35 dish, culture cells at 80 to 90% confluency. Transfect the cells with the plasmid of interest using lipofectamine 2000, as per manufacturer's instructions. Then place the cells in an incubator. On the next day, split the cells into two P150 Petri dishes and incubate overnight. Replenish the media to add 50 micrograms per milliliter of G418. Continue culture under antibiotic selection for 2 weeks. Examine the culture for G418 resistant colonies. Then, using an inverted fluorescent microscope, identify and mark GFP positive colonies. Wash the cells twice with PBS. After the second wash, leave a thin layer of liquid to prevent cells from drying. For each marked colony, wipe as close as possible around the